Hi all, welcome back to Microbiology Lecture. Today we're going to continue talking about eukaryotic microbes, and now we're going to start talking about the protozoans or protista. And so, mind you, like when I talked about this quite a few lectures ago, I was talking about the kingdom protista or protozoan, and I said that to some people, it belongs as a kingdom. To other people, it needs to be split up into multiple kingdoms, or at least the organisms might need to be moved out of the kingdom, some organisms. Okay, we're just going to talk about it in general. The taxonomy of the kingdom protista, just realize that it is ever changing. There's lots of individuals that think that things need to be better to get this to become a monophyletic group, because right now it's a polyphyletic group. But maybe we'll talk about that a different day. So when protozoans were first described, they were described with the term protozoa, which means animal-like, even though we now know that there's lots of protozoans that are plant-like and can do photosynthesis, and there are some that are fungi-like, even though they might not have chitin and some other components of fungi, but they act similar to fungi. So normally when we label something protozoa, we're talking about heterotrophic organisms, although not all the time, and they're not fungi, and so that's normally how we kind of label something as a protozoan. So for an example, like this is red blood cells from a human, and this is Trypanosoma, a species of Trypanosoma. This is a protozoan that gets into the blood of humans and tends to wreak havoc because it consumes blood and can also evade our immune system fairly well. And when we get to Trypanosoma, I'll talk about that a little more. So characteristics of protozoans, for the most part, they lack a cell wall, although there are some photosynthetic protozoans that have cell walls, or at least some organisms that are classified as protozoans that have cell walls. Marine protozoans will often have some type of exoskeleton, and normally that's made out of either calcium carbonate or silica. This is somewhat of a concern when it comes to like global climate change and the warming of the temperatures on the planet. Is also typically associated with the acidification of water, and so because we're increasing the amount of carbon dioxide, we tend to increase the acidity of our waterways, and that can have effects on calcium carbonate organisms. And you might say, well, I don't know that many protozoans, but that doesn't mean they're not really, really important as zooplankton, which we'll talk about here in a little bit. So there are lots of protozoans that occur in the water column that provide a food resource for many organisms, fish, invertebrates, even whales and other organisms that are eating a lot of zooplankton will consume large amounts of protozoans. Many of the protozoans use different features to move around. 3 features that are most common would be cilia, which are like little teeny hairs or very small flagella that are just all over the organism's body or outer surface of their cell, and they can use them to beat and move through the water. And then flagellum, we talked about it before. We talked about prokaryote flagellum. We talked about bacteria having a type of flagella, and then archaea having a different type of flagellum, and now eukaryotes, they have a different type of flagellum. Here you can see these trypanosoma with this flagella structure coming off. And again, that is a great example of what we call convergent evolution. That whip-like tail allows for organisms to move through water or liquid, and it evolved many times because the flagellum of eukaryotes is not related to the flagellum of prokaryotes, and even the flagellum in prokaryotes is not related to each other. And then pseudopods. Pseudopods are extensions of like the body of the organism or the cell membrane of the organism. It's like reaching out a foot and then snaking your body along. Pod or poda means foot, and so pseudo means like fake foot. And we'll look at this when we look at amoebas here in a little bit. So the habitats that they tend to live in, most of them are aquatic, but we can also find large numbers of amoeboids or amoebas in soil. And then often we concentrate on things that might occur in plants and animals that might be parasitic, and we'll talk about those in future lectures. Okay, again, just to harp on that, zooplankton. Lots of organisms eat zooplankton. These are heterotrophic microorganisms that are consuming, typically they're consuming phytoplankton. Phytoplankton meaning organisms that are photosynthetic in the water that are microscopic. The zooplankton would eat the photosynthetic organisms, which are autotrophs, and then the zooplankton is consumed by larger organisms, and that's kind of how a lot of the food cycles go. They can come in all shapes, sizes, structures. There you can see a paramecium that has lots of little teeny hairs, so you can see that those are cilia. Sometimes they occur in colonies or groups, but again, majority are unicellular, so even if they occur in large groups, they could break free and live by themselves. There's an amoeba, and there you can see, they have multiple examples here of the pseudopoda or pseudopod that they use to move throughout the water column. Like we've calculated CFUs for bacteria, and many of you have probably found CFUs, so the number of bacteria in a gram of soil, to be somewhere around this mark also, 10 million or so. And so protozoans can be well established in soil, different soil samples. And what are they doing there? Well, a lot of the amoeba protozoans, they're eating the bacteria and the archaea that occur in the soil. So often they'll be well represented in the soil. Some of them occur in regions and really make a species work. So like the guts of termites have a symbiotic protozoan that lives in the gut of the termite and allows for it to break down cellulose so it can eat wood. And without it, the termite itself could never digest that material and therefore wouldn't exist. So those two organisms interact symbiotically, and so the protozoan can't live by itself and the termite can't live without it. Cattle, kind of same thing, except for normally the rumen of the cattle not only has protozoans in it, but it also has bacteria and archaea, and so they just have a huge community of microorganisms that occur in their rumen that allows them to break down cellulose products into simple sugars that the cattle can then digest. So that's another example of amoeba here that's consumed some photosynthetic organisms, but they can be a large part of the food chain, not just in the soil, but again, back to kind of the zooplankton. They are that group that are often consuming the photosynthetic, the microscopic photosynthetic organisms. They're the microscopic organisms that are consuming the photosynthetic organisms. That's not to say that there aren't protozoans that are photosynthetic. There are, but often they're that intermediate, and so they can serve a large role in food cycling or nutrient cycling. All right, so that kind of brings me to one group, or to talk a little bit in general about intestinal protozoans. These are protozoans that are typically found in the intestinal tract of lots of different species. So normally, because we're mammals, we talk about the ones that are found in mammals, but there are lots of protozoans that are found in birds, and fish, and reptiles, and amphibians, and invertebrates, and a lot of them will have similar life cycles, where they might have a trophozoite that feeds and grows in the intestinal tract. When it gets large enough and it reaches sexual maturity, a lot of times the trophozoites will then go through reproduction, meiosis, produce a cyst, and then that cyst is basically what we call a survival form for the organism. It's like an endospore when we were talking about bacteria. It's like the spores that when we were talking about fungi. The cyst is this survival form that can be defecated out or removed from the body in some other form, sometimes vomited out. But that cyst can occur in the environment, and sometimes that environment is not very favorable to the organism. So it might just be like in water or along the stream bank, and areas that they might not be able to survive, and it can just sit there for long periods of time until conditions are right, like another organism eats it, or it gets into an organism that it then can start that life cycle again. So I'm fairly famous one that causes lots of diarrhea or intestinal issues is Giardia. So this is Giardia's trophozoite form, and so again feeds, multiplies in the intestine. Sometimes there's very little sexual reproduction and a lot of asexual reproduction, but again they're typically going to reproduce as trophozoites, and then when conditions are right to form a cyst, they're going to encyst and then exit through fecal material often. Okay, so in the case of Giardia, they exit through fecal material normally into a water system, or that's a good way to get Giardia, is to pick it up from a water that's been contaminated. And then that cyst survives, and then when a host consumes the cyst makes it to the environment in which, normally in the case of intestinal protozoans, into the intestines, and then once it's into the stomach or intestines, the stomach will often trigger with the acidity of the stomach triggers the cyst to exocyst, breaking open and letting the trophozoites out again so they can start reproducing again. And so this is a cyst form of Giardia. Okay. The amoebas, again, there are amoebas that are medically significant, some of which, if you get them, and we'll talk about Fowleri here in a second, it's an extreme, it's a bad deal if you get it, because we're not its definitive host often, and so some of these things, maybe they don't have a definitive host, or we're not their definitive host, and so sometimes they can wreak havoc on our body. But amoebas, when we're talking about them, again, they have a pseudopod that allows them to move forward, but also engulf particles, so they phagocytize, they wrap their pseudopod around things, push it in, and then consume the material by using digestive enzymes. Most of the time they're reproducing by binary fission, and then like some intestinal amoebas will form a cyst, just like the other protozoans form a cyst, and that is kind of that survival method for them to carry on. So here you can see a cyst form of the amoeba, and then like when that cyst is ingested, just like many other species of protozoan, once it's ingested, it's in the right region, then it exocysts and starts reproducing again. Interestingly for amoebas, which is kind of interesting, is they don't have a mitochondria, and so they rely purely on fermentation. So sometimes with amoeba infections in humans and things, you have this delayed effect, you have this delayed period of time until symptoms come on, because the populations are low at first and fermentation has to occur, and the fermentation process sometimes takes a little bit longer to generate enough energy for them to replicate a lot, and what you can have is kind of a delayed effect. And so here you can see that some amoebas also consume red blood cells, so it's not just consumption of nutrients from the intestines. Sometimes they'll eat red blood cells and feed on other parts or other regions of the body, and so most are not detrimental to humans when humans get them. Sometimes they'll be pathogenic in the sense that it'll cause diarrhea, but again, if you're already if your immune system's already compromised, then they can cause a great deal of distress to the organism. One, though, regardless of the immune system, is Naegara fowleri. This one it's a strange species in the sense that it's super rare to get fowleri. It can enter the body in a few ways. It can go through the nasal passage into the bloodstream. Sometimes if you have a cut or a wound, it can enter through that wound, and there are some limited pieces of evidence that suggest that maybe drinking contaminated water can get it. But fowleri will often move from like the sinuses or the blood and enter the blood-brain barrier and then enter the brain tissue and destroy brain tissue. And depending on the estimates that you read, it's above 95% fatal when people get fowleri. And so often this is, of concern because of the high degree of virulence from this species. Okay, the apicoplexans or the sporozoa, these guys are normally distinguished by the apical complex, which is one end of the parasite will have this drill-like or apical complex structure, so it's kind of like a drill that allows them to enter host cells. Normally, if they're entering host cells, they're consuming some material inside that host cell, so something like Plasmodium would be a good example of a species that's going in. The Plasmodium malaria is going into red blood cells, consuming hemoglobin, and it's that apical complex that allows for them to enter. Now we start seeing in the sporozoas, you start seeing that you have intermediate hosts and definitive hosts. A lot of the amoeba parasites, there's no intermediate host. It's just a definitive host, or they're just a free living organism and they actually don't have a definitive host or an intermediate host. They're not parasites in that sense. They just happen to be opportunistic when they enter an organism. They can wreak havoc on it, like Fowleri. But at any rate, the sporozoas, they have a definitive host and an intermediate host. And a lot of these other pathogens, same deal, like human pathogens, where we often will call them human pathogens, but I don't want you to really think of them as just being human pathogens, because the intermediate hosts can often also be like a definitive host, especially with things like Cryptosporidium. We now know that Cryptosporidium can affect lots of different organisms, and some of which can have the effects of Cryptosporidium. Some are intermediate hosts and they just hold the Cryptosporidium, and then the definitive host is us. But often Cryptosporidium is a digestive disorder from drinking contaminated water, or eating food that was watered with contaminated water. Sometimes this happens, but nonetheless, this whole group is really separated because of this apparatus that occurs on one end of the organism and allows them to enter the cell. When they enter the cell, they will typically consume material in the cell. And sometimes if that's like a red blood cell, then it might be hemoglobin. In other cases, maybe they're consuming stored carbohydrates or proteins or other things that might be inside that cell. So we'll come back and we'll talk about Plasmodium in a little more detail next time.